This video provides an introduction to the basic structure of chromatin and its functions. Generally speaking, chromatin is the complex of DNA and its associated proteins that is found in the nucleus. Sometimes, in textbooks and scientific articles, you will find descriptions of chromatin that differ slightly from this definition. For example, the second definition on the slide also includes chromatin-associated RNAs as integral part of chromatin along with the DNA and its associated proteins. In our class, we will use the first definition. But please remember that there are RNAs such as exist that coats the inactive X chromosome in female mammals that stably associate with chromatin. Chromatin has two main functions. It compacts DNA and it controls access to the information encoded in the DNA. In each nucleus of a human cell, we have approximately 2 meters of DNA. Chromatin compacts this large amount of DNA so it can fit into the nucleus. The various levels of compaction are shown in the figure to the right. The DNA, depicted at the top, is first wound around histone proteins to form nucleosomes, which we will look at in more detail shortly. These nucleosomes are arranged like beads on a string, a model which you are familiar with from your genetics class. The DNA and the nucleosomes are then further compacted to form a 30 nanometer fiber, which again is shown on the right. These first levels of compaction are relatively well understood. Additional levels of compactions occur, as shown on the right, but these are not very well studied. The most compact form of chromatin is present in the mitotic chromosome, which is shown at the very bottom of the figure. During the other parts of the cell cycle, most of the DNA is present in a less condensed form. While it is essential that the DNA is compacted in the nucleus, chromatin at the same time has to ensure that the cellular machinery, such as RNA polymerases, can access the specific regions of DNA that are needed. In eukaryotic genomes, regions of DNA that need to be accessed, such as protein coding genes, are tightly interspersed with regions that need to be kept inaccessible, such as transposable elements. Thus, chromatin has to control access on a relatively fine scale to ensure that the correct sequences are accessible or active, and others are inaccessible or silent. To understand how chromatin carries out its functions, we will look first at its basic unit, the nucleosome. A nucleosome consists of 147 base pairs of DNA wrapped around an octamer of histone proteins. There are 2 molecules each of histone H2A, H2B, H3, and H4. On this slide, you have 2 depictions of the nucleosome. On the left, there is a cartoon diagram showing the makeup of the nucleosome, and on the right, you see its molecular structure. Generally, DNA is negatively charged. Histones are rich in lysines, leading to an overall positive charge. There are many contacts between the DNA and the histone proteins, and because of their opposing charge, they closely interact. Please note that the histone molecules have N-terminal tails that protrude from the nucleosome core particles. The histone tails are extremely important because they can be post-translationally modified and thus influence the interactions between the DNA and the histones and between the nucleosomes and other chromatin proteins such as transcription factors. Nucleosomes are assembled in a stepwise fashion with the help of chaperones, a process that is depicted in this figure. First, 2 types of dimers are formed. 1 molecule each of H3 and H4 form one dimer, while 1 molecule of each of H2A and H2B form the other type of dimer. Next, 2 H3-H4 dimers form a tetramer. To this tetramer, 2 dimers of H2A, H2B are added to form the histone octamer that is now ready to interact with the DNA. At various points during the life of histones, either before they are incorporated into a nucleosome or after, they can be post-translationally modified. While modification can occur at the histone core particles, most of the modifications occur at the N-terminal tails, and some of them are shown here in this diagram. Modifications can include, for example, acetylation, methylation, phosphorylation, and ubiquitination, among others. On each histone tail, there are multiple modification sites, and modifications can occur alone or in combination. Given the number of possible modifications and the presence of 8 histone molecules in each nucleosome, a very large number of differentially modified nucleosomes can exist, all with different impacts on the compaction or accessibility provided by the chromatin structure. In summary, in this video, we have defined chromatin as the DNA and its associated proteins. We have discussed that chromatin compacts the DNA in the nucleus while controlling access to the information encoded in the DNA. We have looked at the basic unit of chromatin, the nucleosome, and learned that it consists of a histone octamer and 140 base pairs of DNA. Finally, we have seen that histones can be post-translationally modified in a variety of ways that impact chromatin compaction and access. We will further discuss these modifications later in this semester.